Nanomedicine is the science and technology of using nano-sized tools for the diagnosis, prevention, and treatment of disease to gain increased understanding of the underlying pathophysiology with an ultimate goal of improving quality of life. COVID-19 pandemic is one of the biggest examples of the significant impact that nanomedicine has had on all of our lives. Nanomedicine was employed during the various stages of the research related to COVID-19. It played a vital role in the development of the COVID vaccine, which has ultimately proven its importance in controlling the spread of this virus. One of the most prominent COVID vaccines is the mRNA-based vaccine. mRNA is a genetic material that instructs our cells to produce proteins such as antibodies. With nanotechnology, mRNA is encapsulated in a layer of a lipid nanoparticle that protects it and helps it to get taken up by specialized immune cells called dendritic cells. The biocompatible and biodegradable nature of the lipid nanoparticle mimics our cell membranes and allows for the entry of the mRNA inside the cell to do its job as it safely disintegrates upon entering. Aside from the development of vaccines, nanomedicine is a valuable tool for drug delivery research, antimicrobial, diagnostic, and imaging tools, wearable devices, implants, screening platforms, and so on. Many of which use nano-sized biological, non-biological, or hybrid materials called nanoparticles that encapsulate therapeutic agents. Different nanoparticles with various sizes, shapes, and surface chemistries are now available for many biomedical applications. However, the therapeutic efficacy and fate of these nanoparticles are heavily influenced by these different properties. Nanoparticles can be engineered to obtain the desired properties such as biocompatibility, physiological stability, selective targeting, increased drug loading capacity, and the ability to carry both hydrophobic and hydrophilic drugs. Nanomedicine has the potential advantage of overcoming many barriers that are witnessed with conventional medical practices. For example, in cancer therapy, nanoparticles can significantly reduce the side effects associated with conventional drugs in two ways. First, due to their extremely small size, they can enter the tumor sites because of their localized leaky vasculature and are retained in their place because of the poor lymphatic drainage in the tumor environment, where they can release the therapeutic anti-cancer agent. This is known as the passive targeting approach. Another approach is active targeting, where nanoparticle servers are modified with targeting ligands that will only attach to diseased tissues, sparing the healthy neighboring cells and reducing side effects. Among the most exciting advances in nanomedicine are smart or multifunctional nanoparticles that use a theranostic approach where they can simultaneously diagnose, treat, and even monitor treatment efficacy. These smart carriers are engineered to provide contrast at the zone of interest and give information about the local environment, so this information can help in imaging the anatomical fine structures of organs and identifying tissues with specific biomarks, as well as enabling local interpretation of the concentrations of molecules of interest. This aids in disease analysis directly in the human body while also being used as delivery vehicles for pharmaceutical agents and provide point-of-care therapy in a non-invasive way. There are many types of smart nanocarrier systems. They are either organic or inorganic in nature. Some of the most commonly used organic nanoparticles are dendrimers, solid lipid nanoparticles, micelles, and liposomes. While the most commonly used inorganic nanoparticles are metal and metal oxide nanoparticles, quantum dots, carbon nanotubes, upconversion nanoparticles, and mesoporous silica nanoparticles. Nanoparticles can also be protein-based that are used as targeted drug delivery carriers, or viral-like, which have many medical applications for the treatment of viral diseases. Quantum dots are a type of inorganic nanoparticles that are widely used for the treatment of a variety of diseases. They range in size from 2 to 10 nanometers. Their favorable chemical and physical characteristics, such as their unique optical properties and fluorescence emissions, have sparked an interest among researchers, leading to the development of various applications in biosensing and bioimaging. Due to their unique size-related physical, chemical, optical, and biological characteristics, gold nanoparticles have also been widely investigated for a variety of medical applications in drug delivery and bioimaging. Their properties make them ideal non-toxic carriers for drug delivery applications. Drugs could be conjugated to the surface of gold nanoparticles by forming covalent bonds with the help of sulfur atoms. It was demonstrated that gold nanoparticles could carry the drugs and enter cells of various types like lymphocytes, macrophages, and brain microendothelial cells where HIV is known to replicate and consequently effectively inhibit HIV replication, making them a suitable option for the treatment of viral diseases. Because of their localized surface plasma resonance, inorganic nanoparticles exhibit strong light, scattering, and absorptions in the visible and near-infrared regions. The absorbed light is then converted into thermal energy. Scientists have applied this mechanism to cancer therapy, where inorganic nanoparticles conjugated to antibodies are recognized and taken up by tumor cells. Organic-based nanoparticles are one of the largest groups of nanomaterials with a great number of biomedical applications due to their biocompatible and biodegradable properties. Phospholipids with hydrophilic heads and hydrophobic tails are the building blocks of liposomes, which are nanoparticles with a lot of structural similarities to cell membranes. When phospholipids come in contact with an aqueous environment, they self-assemble in a way that makes the hydrophobic tails point towards each other, while the hydrophilic heads seek the aqueous medium. This arrangement allows for the formation of a phospholipid bilayer around an aqueous core. The lipid bilayers seal off to form a liposome. Different drugs can be incorporated in this structure for the safe delivery of therapeutic agents. Liposomes are now one of the most advanced clinical translation delivery vehicles, with many formulations licensed by the U.S. Food and Drug Administration, and liposome-based drug encapsulation systems are already being employed in some cancer therapies. Dendrimers are globular nanostructures with repeated layers extending from an initiator core. They can be modified with surface terminal groups that resemble tree-like architecture. These structures have been investigated for use in a variety of biological applications, including drug, DNA, and protein delivery, as well as bioimaging. Nanomedicine is a rapidly progressing field, and the research that has come so far is beyond anything anyone has dreamed of years ago. For example, nano-sized machines or nanobots are already being developed for medical applications. In such plans, future medical nanotechnology is expected to employ nanobots injected into the patient to perform work at a cellular level. However, the safety profiles of the nanoparticles are not well established yet, with some showing signs of toxicity in the early stages of development. Nanomedicine, like medicine, can enter clinics and become a part of standard clinical practice if all aspects of translations are met, including safety, regulatory, and ethical standards. So ideally, nanomaterial development should incorporate a safety by design approach to ensure these standards are met, with an ultimate goal to improve patients' quality of life.